Hey guys, so today we're going to be talking about the Golgi apparatus. So in our last video, we started the basics of the endomembrane system, including the smooth ER and the rough ER, and a lot more detail about ribosomes. And one of the things we talked about with ribosomes last time are the way that the ribosomes decide if they're going to be free in the cytosol or bound to the rough ER. So we remember, hopefully, that proteins that need to stay in the cytosol, see these guys here, or travel into the nuclear space, be synthesized by a free ribosome, while things that need to be in the endomembrane system somewhere, so they need to stay there or sort there, something about needing to be in the endomembrane system, proteins that are embedded in the membrane, or proteins that are secreted from the cell will need to be translated by a bound ribosome into the lumen of the rough ER. Okay, so now that our protein is in the rough ER, what do we do? The next step is to travel to the Golgi apparatus. So here's the Golgi apparatus, and you may remember this as being referred to as sort of the post office of the cell, because it's responsible largely for sort of sorting out and sending out proteins to where they need to go, okay? The Golgi apparatus is effectively a network of membranous stacks, kind of pancake style, that we refer to as cisternae. So here we've got the cis face of the Golgi. So this is going to be the face of the Golgi that's closest to the rough ER, and the trans face of the Golgi, which is going to be the face of the Golgi that is closest to the plasma membrane. So generally speaking, things enter through the cis face, exit through the trans face. Okay, so when proteins are ready to leave the rough ER, they're packaged in what are called vesicles, which are basically small bubbles of membrane with proteins encased inside. So we'll ultimately be learning more about how these function, okay, and how we know what to package in a vesicle. These vesicles enter the cis face of our Golgi apparatus. Okay, once they enter, they're modified, sorted, kind of picked out where they need to go, and a new vesicle will leave at some point through the trans face, maybe even one of these later medial faces of the Golgi. Okay, so generally that's how we're going to be rolling through the Golgi apparatus. Okay, as these proteins travel through, again, they're sorted and sent to their destination. Carbohydrates and other molecules are added. So the Golgi is, yes, the sorting organelle, but it's going to do so through kind of adding in some of these other modifications like carbohydrates, lipids sometimes, things like that. These extra molecules can be part of how the protein ultimately needs to function. It can help them know where in the cell to go, which is kind of what we'll be focusing on today, or help them maintain their overall 3D structure, that sort of tertiary or quaternary structure. Okay, so let's take a closer look at the ER and the Golgi apparatus. Okay, so now we have, again, our rough ER. Closest to the rough ER, we have the cis face of the Golgi. Furthest from that and closest to the plasma membrane, we have the trans face. And in the middle, we have the medial face. Okay, so we've got cis, medial, and trans faces. All right, so zooming in a little further. Before we start adding in proteins to this model, it's important to understand generally how the Golgi apparatus is going to operate. So in this sort of idea of the Golgi, the cisternae and their contents are going to make their way from cis face to the trans face. And this is going to be via what we refer to as the cisternal maturation model. Along the way, once we do add the proteins back in, certain types of lipids, we'll modify them and sort them, okay? So what do we mean by cisternal maturation? What we really mean is that each of these cisternae kind of more or less stays together and will slowly make its way from the cis face all the way to the trans face, okay? So let's see how that works. As things finish up in the Golgi apparatus, they'll leave from vesicles from the transphase. So we just saw that happen. The next Golgi cisterna will come in and take the place of the transphase. It becomes now the new transphase. So a piece of medial Golgi next in line forms the new transphase. The guy behind it scoots up, moving forward. So ultimately, as you can see, each of these cisternae will have its turn to be the transphase. So what happens when we get to the cis phase? New vesicles from the ER will come in and form the new cis phase. So we have this sort of continuous sort of treadmill, conveyor belt of cisternae. Okay, so now let's zoom in even a little bit more and look at some things that are in the Golgi. Okay, so of course all types of proteins are going to be in the Golgi. So here's some different types of proteins, all of which were synthesized by a bound ribosome. So they all have a signal peptide in there somewhere. Okay, so as the cisternae mature, the proteins will come along with it. So the proteins that were initially part of the cis phase, as the cisterna matures, it will become part of the medial Golgi and all the proteins with it. Okay, so let's keep going, moving along, another round of maturation. As we travel through these cisternae, carbohydrates and other molecules can be added to our proteins. Sometimes they don't have to be, but here are some carbohydrates that I've added. Okay, so these are sugars, just carbohydrates. When these sugars are added, this is called glycosylation. And the resulting protein is usually what's called a glycoprotein. So it's a protein with a sugar element like glycoprotein. Okay, so we've now got our glycoprotein, carbohydrates added. Here's another example down here. Okay, so we're moving right along. At this point, you can see some of the molecules may leave from the medial stack, sort of at the ends, while others can be modified further. So here's some additional carbohydrates, some more glycosylation happening. Go. Moving further through, some of those little heart guys have left completely now. The rest of the guys are waiting to make it to the transphase. Once we're there, we can exit in vesicles. Okay, so those carbohydrates that we've added, in some cases, can help us sort the protein. So it's kind of like adding an address onto the proteins, just as you would for the post office. We have not seen how that takes place just yet, but in subsequent videos, we will get some examples. Okay, so just in conclusion, the Golgi apparatus is an important organelle involved with sorting. So a lot of the things that have to go through the ER need to ultimately make it to the Golgi so they can be sorted and sent to where they need to go. And along the way, these proteins are modified with carbohydrates and other functional groups. Sometimes we add things like phosphates, which we'll see examples of. These carbohydrates definitively help with sorting. We'll get some good examples of that. They also can help form structure and participate functionally in the final product. They need to be there so that the protein can function. Next time, we'll see some examples of how this works.